We will continue our discussion about spike protein from the SARS-CoV-2 virus when it is expressed in different cell system, how the glycosylation profile changes. So in our previous video we have discussed about O-glycosylation, and we will continue our discussion from where we end in our previous video. So in the case of spike proteins that are expressed from HEC293 cells, what has been observed is that it has 9 O-glycan site, whereas the spike protein that is expressed from BCL2 cell system has 16 O-glycan sites, and most of the O-glycans in the BCL2 are fucosylated, whereas in HEC292 293 spike proteins from HEC293 cell lines expressed spike protein has some of the sialylated glycans, as it can be seen here. These are the sialylated glycans, and this is expected as in insect cell lines, that is Baculovirus insect does not have the capability to form sialylated glycans. That's why we do not see any sialylated glycans in the spike proteins that are expressed from BCL2 cell system. And in this figure we can see that the glycans that are similar are marked with the same color, as it can be seen. For example, S371 in HEC93 has same glycans as that of spike protein that are expressed from S371 from BCL2 virus, and the area in the circle represent the abundance. So this T478 has more abundance, and S477 also has more abundance compared to the other cell system, and similar is the case with the HEC293 cell system. This T478, TS477 and S498, and in case of BCL2 S494 also has more abundance compared to the other cell system. So this is how we can interpret this diagram. The same color represents the same glycans in the different cell system, and the area of this glycan within the circle represent the relative abundance within the in the cell system. Later on, they decided to check whether this O-glycan site has any contribution towards its SARS-CoV-2 attachment or this virus attachment with the ACE2 receptor. So they have first identified 10 ribosome these 10 binding motifs of the SARS-CoV-2 virus, and 10 receptor binding motifs of the SARS-CoV-2 virus and the ACE2 receptor, and they checked whether there is a O-glycosylation site exists at these sites or not. And these sites are shown in this panel A. As we can see there, they are marked as at site 1, site 2, site 3, site 4, and continue to the site 10. And what they have found out, one of the thing is that before each site, or at before after the before adjacent to the each site or within the site, it contain either amino acid T or amino acid S, as it can be seen here. That is, in the case of site 1 it has the S amino acid. In the case of in the case of site 3 it also has S amino acid. So this is this observation is valid for most of the sites, but not for all sites, as in site 2 there is no S or T. So from this from this major availability of S and T to the neighboring site, they made a point that it could be the O-glycans that are playing a role in the ACE2 receptor and virus binding, as O-glycan sites are usually attached to S and T sites. So that then later on they decided to check whether this receptor binding domain has either O-glycan site or N-glycan site in abundance. And when they check for SARS-CoV-2, they can see that it has one N-glycan site, whereas SARS-CoV-2 has two N-glycan site and few O-glycan sites also. Later on, they go more in detail and to check the sites that are observed to have play the role in the receptor binding domain and ACE2 receptor, whether there is an glycosylation, whether there is a O-glycosylation site nearby to that or not. So they check three sites, that is site 1, site 7, and site 9. And in the case of site 1, they can see that O-glycans are observed near S438 point, whereas there is no O-glycan for SARS-CoV. And similar is the case with the site 7, where they can see the O-glycan at QS at 494 uh, position, as it can be seen from the purple color, whereas there is no O-glycans for SARS-CoV. And for the at site 9, they can see the O-glycans both for the SARS-CoV-2 and also for the SARS-CoV. And SARS-CoV, they can see the O-glycans at two similar position, that is at 485 and 486. And for the SARS-CoV-2, they can see the O-glycans at this 500 position. And different kind of O-glycans are present at this position, similar for the site 7 and uh, site 1. So from this they made a point, or they come to a point that O-glycans might play a role in receptor binding motif for the SARS-CoV-2 virus more compared to the SARS-CoV. That's why SARS-CoV-2 is more infectious compared to the SARS-CoV. They made uh, that could be one of the point that why SARS-CoV-2 is more infectious compared to the SARS-CoV. So their observations seems like in line with their result seems like in line with the observations that SARS-CoV-2 is more infectious compared to the SARS-CoV. And one of the reason for that this study shows is that O-glycosylation near the receptor binding motif might helps in the attachment of helps in the this attachment of SARS-CoV-2 virus to the ACE2 receptor on the cells. So I hope you have understood some of the basic understanding of how this O-glycosylation helps SARS-CoV-2 to binds with the ACE2 receptor. If you need to understand these things in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.